I apologize. I am a little jet lagged. I've just got back to town after taking care of my parents who were both ill, and many of the things, believe it or not, that Deneen said, my father actually has Alzheimer's. You'd be amazed at how many of the things you said are so universal to other health problems as well, and it was wonderful hearing it. Thank you very much. I'm going to be talking about hope, hopeful biomarkers. We are doing research at the University of Utah, and we are trying to come up with biological hard science biomarkers so that no more of this skepticism and all in your head noise will be part of the future of fibromyalgia. That's at least one of our hopes. And the second part of this hope is if these biomarkers give us enough of a clue, we may even see the beginnings of places where we can go for innovative new treatments, and that's down the road. I mean, but I do want to emphasize that what I'm going to be talking about here is research. So some of what I say is so new that you have to put it in the perspective of give it time, and we'll see if this all pans out. So that's the scientist in me. I have to say that. Okay. Okay. This is at least the largest part of our research team at the University of Utah, and you can see the name at the top has the same last name as mine. And this is because it is my husband, and he is the actual innovator and true believer. He is the one who found these sensory pathways that I'm going to be talking to you today about. And he found them, for those of you who are skeptics about animal research, he found them through animal research. And don't put that down. It's very, very important that we have that side going as well as the human research, which is what I do. Myself, Ron Hugan, who is our super tech, who really, he makes the project work. He's the one who has the technical skills to do this science very well. Andrea White, who is our exercise scientist, and she's a very active member of our group. And then one of our later speakers and our organizer, Cindy Bateman. And I just want to say that I was so disorganized after my trip that I didn't come prepared to recruit those of the audience who might be interested in being volunteers for our study. So I'm hoping that you'll go to Cindy's booth, which is just to the left outside that door, and sign up for her studies in general, and then we can hopefully get your name passed on to us if you are interested in participating. Okay, so everybody in the room probably knows about fibromyalgia syndrome and its definition better than I do. but I keep this slide going mostly because I like to point out that the thing that you heard over and over again, that this is a pain syndrome, and more than that, it's very widespread pain. All parts of your body, bilateral, upper and lower, and that is the central characteristic. The second most common characteristic, as you already heard, is fatigue. And so we are studying actually both the pain and the fatigue side of this syndrome. Now, the definition, though, for the clinicians is based on hypersensitivity to pressure at tender points on the body. And this is something, doctors and physicians love things that are quantitative. So we end up with this, you have to respond in a certain way to 11 of 18 tender points when somebody pokes you with their finger. And so what if you respond with 10 or 9? You don't qualify. Too bad. You didn't make the quantitative threshold. It's really a bit of a pain that you deal with these things in these kinds of numbers. And certainly for patients it can be very frustrating because, as we know, this is a cyclical disorder. So on Thursday you're at 18. and the following Thursday you're at 9. Does that mean you don't no longer have fibromyalgia? No, of course not. So anyway, the numerical side of this, which is, you have to be aware of the medical models that we're working with here, is one of the reasons that we're looking for other biomarkers and not just sticking with the old definition, which is still very helpful if you want to get insurance coverage. So, okay. Now, the other thing that on this little note is the bottom line here. In our subjects, up to 70% or more of the individuals that we see also meet criteria for chronic fatigue syndrome. And in fact, when we do our studies, it doesn't seem to matter a whole lot. They come in with a diagnosis, whatever was their first diagnosis, whether it was fibromyalgia or chronic fatigue syndrome, but then we test them to see if they meet criteria for the other one, and it definitely seems to go both ways. That the ones who only knew they had chronic fatigue, still the majority of them meet criteria for fibromyalgia, and vice versa. And that is because fatigue is the second most dominant symptom in this syndrome after pain. Okay, so this is the kind of picture that we have about fibromyalgia, that it has to do with muscles and ligaments and tendons, and then if you break the muscle down, those are all the parts of the muscle, the surrounding fascia, and going all the way out to the myofilaments of the muscle, and then those sensitive points all over the body. So this is something that I just simply lifted from the internet as an image of fibromyalgia. And the only reason I look at that is all about pain. And again, we are actually focusing on the fatigue component just as much as the pain component. And if you think about muscles, they do fatigue just as they can feel pain. So again, here's the other half of our syndromes of interest in our research, chronic fatigue syndrome. And again, for those of you who have it, you're probably familiar with this. For those of you who've only been diagnosed with fibromyalgia, you might want to think if you actually meet this set of criteria as well. Do you have fatigue lasting more than 6 months that it severely impairs your function at least half of the time? And do you have, again, this is another of these very difficult medical definitions. You must have 4 of the following 8 additional symptoms, and I'm not showing all of them. I'm only showing the key ones for our project. Muscle pain, joint pain, and symptoms worsen 24 hours after physical exertion. So there are others as well, but the notion that you have 4 of 8, again, we're into this numbers game. What if you have 3 of 8? You know, again, it really speaks to the need for a hard, medically accepted biomarker for both of these disorders. My husband, who is the true neuroscientist in the family, wants me to also include this notion that genes must very likely be involved in both of these disorders, and certainly are involved in how everybody experiences pain and experiences fatigue. And most of us are very aware of genes in terms of this first component, the ones that have to do with the kind of genes you inherit from your parents. And we think of that as kind of a fixed thing. You know, we either got it or we didn't get it from our mother or our father. We can think of that in relation to these disorders as something where you would inherit in these genes a kind of constitutional vulnerability, something that might make you more susceptible, and you may not even know you have it until something happens. However, genes are also something that is not fixed or immutable. In other words, what can happen is something in life, a life event, some kind of infection, something like that, a traumatic accident, a fall, a major life stressor, you get divorced, you lose your job. those things can influence the expression of genes. And by that I mean it can turn on specific genes so that there start being more and more and more replications of what these genes actually do. So you can turn on that gene expression due to some environmental event. Think about genes in both ways. The more immutable one where you're inheriting a vulnerability, and then the side that has to do with gene expression. We also, in our research, we are focusing on specific short-term challenges like exercise or like a short-term stressor that we can put people through, and those can alter gene expression, something that you might not have known. And that is actually what we look at as the measures in our study. Now here is a real headliner for those of you who thought that scientists were so smart. Scientists did not, until very, very recently, have even much of a clue what sensory pathways were involved in the sensation of muscle pain or muscle fatigue, even as experienced by healthy people. They didn't know. And yet we knew that what are the two primary reasons people go to the doctor? The symptoms they report most often? I feel pain, I feel fatigue. Those are it. Number one and number two, and yet we didn't know. We didn't know. Pain from skin damage, like cuts or poking or pinching the skin. That has been studied a great deal over decades, and the nervous system pathways about that is pretty well known. And my husband worked on that for a very long time. Pain from working muscles that are not obviously damaged, no actual damage that we can pick out when we look at it, is something that you and I, everybody, even those people who are not fibromyalgia patients in the room, we've all felt it. We know what that feels like, muscle soreness. And those pathways are just recently beginning to be documented. And again, that's mostly come from animal model research, something that we're going to benefit from. Likewise, we've all experienced muscle fatigue. Who hasn't? I think everybody in the room can say we've worked our muscles sometimes to the point where that muscle tells us, oh man, I don't want to do that again. And like pain, fatigue is a vital protective sensory experience. I mean, we have it because it's probably going to save our life someday. That's the whole reason we have these kind of adaptive processes going on. But what can happen is that the body will, because it is protective, have perhaps a tendency to move too far in that direction, and at times when it's not going to really affect you at all in terms of saving your life, it's going to turn on and give you that message, stop, stop, stop. So, okay, one of the things that is an example of this is what's called delayed onset muscle pain. If somebody goes out and uses a muscle that they haven't built up, but they're thinking, okay, I'm going to be the weekend warrior and I'm getting myself back in shape and I'm not going to do this gradually, I'm just going to go for it. 24 hours later, what happens? Ouch, ouch, ouch. Right, okay. And that's a familiar phenomenon. So what happens in fibromyalgia syndrome and chronic fatigue syndrome? Well, here's this paradox. Gradual increases in whole body exercise is one of the most effective treatment regimens to slowly reduce symptoms and gradually normalize function. Yet even moderate levels of exercise can cause the same kind of delayed onset muscle pain. For fibromyalgia patients and chronic fatigue patients, they don't have to work at an excessive level, a mild level, a moderate level. 24 hours, 48 hours later, they feel delayed onset muscle pain, and it makes all of their symptoms worse. And this is one of the, what we call it is this payback. Yes, I want to do that. I know that over the long term it's going to make me better, but I know I'm going to pay. I'm going to pay if I do. And so this was one of the inspirations for our research project. Can changes seen after exercise be one way to get at hard biomarkers that we can use to define chronic fatigue syndrome and fibromyalgia? That's what we wanted to see. And partly during the same time, this study that I'm going to tell you about has been going on for 2 years. But about a year ago, there was a very interesting report from another group from the University of the Pacific. And I don't know if probably some of the people in this room attended some of the conferences where this was first presented. This was another bicycle exercise protocol, which is the same kind of protocol in general that we use. And in this protocol, what they did is they exercised chronic fatigue patients basically to exhaustion, but at least to 85% of their maximum heart rate. And then they repeated this exercise test 24 hours later, right during that time frame when normally chronic fatigue patients feel terrible in terms of that delayed onset muscle pain and delayed onset fatigue. So what they looked at these very technical things that had to do with peak oxygen consumption and so forth. It's very scientific. But the key point is that healthy normal controls, when retested 24 hours later, showed only about a 2 to 3% drop in their oxygen consumption with the second test. But chronic fatigue patients declined by an average of 22 to 27%. Much different, much different. Based on the premise from other research that the second day change from the first day should not exceed 8%, this particular study indicated a significant impairment in metabolic capability 24 hours after the initial exercise test. So they were hoping, these scientists were hoping that these kinds of measures on a test-retest basis could be used as a biomarker itself of chronic fatigue syndrome, and beyond that could perhaps be used as evidence when taken to court in disability cases that this person literally does have a concrete, identifiable inability to work in that time frame, that there is a metabolic factor that is determining their limitations, not something in their head. Okay. Well, although the test-retest, there's probably they're going to continue to pursue this. I know several labs now that are pursuing this model. This was done in just 6 patients and 6 controls, which makes it a very preliminary finding. I know there are other labs now that are continuing with this model. But in our case, we wondered if we could, instead of retesting people 24 hours after exercise, could we look at their blood 24 hours after exercise and find something in that would give us the same kind of information and in terms of identifying which individuals were impaired functionally. Okay, why are we looking for blood-based biomarkers? Well, as I said, the tender point exam, which is very helpful, very important that they accept this medically. But it still is this numeric thing where one day you might meet criteria and another day you might not. And also, it is a self-report measure. The person has to say, ouch. That hurts. Any self-report measure, if someone wants to be skeptical and say, you're a malingerer, you'd say that anyway. Whatever. It can be viewed as something non-objective and potentially biased. The other factor is that blood-based diagnostic measures are the traditional measures in medical practice. They are the kind of measures that doctors are used to seeing. They put it in the file. Oh, look at that. I believe that. And it's considered objective, hard evidence. So it avoids legal concerns, and it has this persuasibility to physicians, maybe to family, maybe to anybody that you want to convince that this is a real disorder. And again, avoids all this subjective and potential bias. So, Alan and his research team just published a report in animal models trying to define these previously unknown or unverified, let's say, sensory pathways that are involved in muscle fatigue and pain. And in his studies, what he found was that it required 2 or even more types of these specialized receptors. They are molecular receptor, little tiny things that are ion channel receptors. And 2 or more different types must be activated together simultaneously to detect muscle fatigue and pain. And what do they detect? Well, in his research, it looks like they detect acid, especially lactic acid, but that's not enough by itself. They also have to, at the same time, be detecting changes in adenosine triphosphate, ATP. We all heard that in our old science classes. I know you know what ATP is. So they have to detect ATP also. Now, what happens in terms of muscles? Well, when muscles are active, they will probably produce some amounts of changes in pH, that's acid, and especially lactic acid, and changes in ATP. Even when you're not doing very much, even when you're just walking around a little bit, some of that will be produced. But when you really work hard, they'll produce more, and they'll produce more, and they'll produce more. And the way these receptors work is they actually function in such a way that they respond differently if they're low levels and than if there are really high levels. But we think that if the system responds abnormally, they can respond to low levels just as if there were high levels. And we think that might be what is going on when something happens to the genetic expression of these receptors so that all of a sudden they multiply like crazy and you have way, way, way too many of them. So that's what we think can be going on here. So in our study, we wanted to know, can we determine if one or more of these ion channel receptors is functioning abnormally in chronic fatigue syndrome and fibromyalgia syndrome? And how did we go about doing this? Again, I'm getting back to the blood. We wanted a blood-based measure. White blood cells, also called leukocytes, are an interesting target for this for a lot of reasons. First of all, we know already, we've known for a long time, that immune and inflammatory responses involve actions of these white blood cells. And inflammatory responses, we think, are involved quite directly in a lot of types of chronic pain. Also, these white blood cells express receptors of many kinds. In other words, express, okay, that means there's at least some of them there all of the time, and they can be stimulated to produce a lot more of these receptors so that, in other words, they can express more in response to changing conditions. And some of these receptors include those key ion channel receptors that appeared in Al's animal research to sense muscle pain and fatigue. So they do actually express these key receptors, the ones we really think are involved. The other nice, interesting thing about white blood cells is when you're dealing with a disorder that involves pain all over the place, very widespread pain, very widespread fatigue, they're carried throughout the body. They have contact with all tissues, including all the muscles, all the connective tissue around the joints, and they have contact with the nervous system, including the brain, which also has these same receptors present. The brain. So for those of you, when they do say it's all in your head, I mean the brain is in the head. That's not. We can look at the brain too. Okay. Now, when a series of blood samples are taken from the same person at different time points, one of the things that we found in our very early research is that the numbers of the ion channel receptors on their leukocytes, on these white blood cells, can increase. And they can increase in a time frame that seems to go along with when symptoms get worse, in a time frame like 24 and 48 hours. So again, getting back to what we believe may be going on here, and I say we, Al believes this, and he's really made me believe this. We believe that very large increases in these receptors could lead to sensations of muscle pain and fatigue even when the muscles are not working hard, even when you're sitting still in your chair, like right now. Okay, now this is just to give you a picture. We wanted to do whole body exercise because we wanted to stimulate receptors all over the arms and the legs and the chest and the neck in our exercise protocol. And of course the blood will go to all those areas, have contact with all of them. So we use this. This is an Airdyne bicycle. What you do with it, you pedal away, work your butt, work your legs, work your calves, but at the same time you do this with your arms. So you work forearms, shoulders, neck, even your pectoral area with this. So it's really quite active whole body exercise. But we wanted to set it at a level of exercise because we know that normal people can experience delayed onset muscle pain too. So we didn't want to make the exercise challenge too strenuous. What we wanted to do was hit a threshold with mild exercise where we knew that our patients would feel bad the next day, feel worse, but probably the normal controls would feel nothing. It would be it's gone. So we did not use the same level of exercise as in the Van Ness study that I talked to you about before. Now, again, we were fortunate, by the way, in such very difficult funding times from NIH, we were able to get a small 2-year NIH pilot study grant to do this. And in this study, we used 5 different time points when we sampled blood, starting before the exercise task and running up to 48 hours after the exercise task, to look for these markers, these ion channel receptors. At the same time, we looked at a couple of other things that we thought were potentially interesting. We looked at the sympathetic nervous system receptors because they influence blood flow to your muscles. And that was something that I've studied a lot during my research career. And we also looked at cytokines. And for those of you who don't know, cytokines they are factors in the body that can be involved in both making inflammation worse, pro-inflammatory cytokines, or turning off inflammatory cycles, and those are anti-inflammatory cytokines. So they're involved in both directions in an inflammatory cycle. So again, the exercise we picked was very moderate, and I'm going to be reporting on the data that we have so far. And again, this is a new protocol, new data. As far as I know, we are the only people to have done anything in this direction in human beings at all, let alone patients with fibromyalgia and chronic fatigue syndrome. So we've looked at criteria for chronic fatigue syndrome. 15 of them also met criteria for fibromyalgia syndrome. Only 5 of them that we studied so far did not meet criteria for fibromyalgia syndrome, but did for chronic fatigue. And I will tell you that some of those would have met criteria for chronic widespread pain, but when you pushed on them, they said, nah, it feels fine. But didn't mean they weren't experiencing chronic pain. They were, and very disabling chronic pain, but they didn't have this hypersensitivity. We also compared their responses to 17 healthy controls. Okay, so we were looking for greater increases in these ion channel receptors on their white blood cells shown by the patients than were shown by controls. And the exercise task was 25 minutes on that Airdyne, which is a long time, but it was set at a mild enough level, which is just 70% of your predicted maximum heart rate, such that even our very, very sick patients, and some of them were very, very sick, every one of them finished the protocol. We didn't have a single one who didn't finish. We were so happy. And then we were going to look at these receptors immediately after exercise, and then later on at 8, 24, and 48 hours after exercise when the symptoms seem to get worse. So again, our hypothesis is the same task. If we're the same very mild exercise task to achieve the same level of heart rate and the same level of blood pressure in healthy controls and in patients might have very different long-term effects in the patients in terms of these biomarkers that we're looking for. Okay, here are our results. Beginning at 30 minutes after exercise and continuing all the way out at 8, 24, 48 hours after exercise, chronic fatigue patients, including those who both did and did not meet criteria for fibromyalgia, increased one type of these ion channel receptors that I'm just going to call type P to 4 times its pre-exercise level. Healthy subjects showed no increases at all. And based on Al's animal research, this type of receptor seems especially sensitive to muscle fatigue. So here's a quick picture of what the group differences are. You can barely see my control group data at all because they're sitting right on 0. Now the time is this, will this? Yes. This is 30 minutes after exercise, 8 hours after exercise, 24 hours after exercise, 48 hours after exercise. Controls, right at 0, right at 0, right at 0, right at 0. Patients, all the way up here. This is the increase from baseline. And you can see this is percent increase. They're really, really up there. And it lasts. So. And then one of these other ion channel receptors, again, beginning at 30 minutes after exercise and continuing all the way out to 48 hours after exercise, those patients who met criteria for fibromyalgia. Now, everybody met criteria for chronic fatigue, but these were just the ones who had pain and hypersensitivity at those tender points, increased one of the other ion channel receptors, which we'll call type A, to 2 times its pre-exercise level. And again, healthy subjects showed no increases at all. This type of receptor in the animal research seems sensitive to both muscle pain and fatigue. And so this is that receptor. And again, if you look here, the controls in the stripes, they're actually a little below baseline here, and very little change from baseline here. And the patients, and again, this is the group that experienced pain and had hypersensitivity at those tender points, really had true full-blown fibromyalgia. They look different. They had dramatic increases in these receptors, especially at 24 hours. Okay, both the chronic fatigue and fibromyalgia patients also showed increases in those receptors that detect sympathetic nervous system activity, adrenergic receptors. And those increases were 2 to 6 times their pre-exercise levels. Animal studies by Al with a colleague, John Levine, have indicated that if you inject muscles of rats with a beta blocker, a beta receptor blocker called propranolol, this will reduce their pain responses during a chronic pain model that they use, which is pain induced by inflammation. And we, in my previous research, when I was back at the University of North Carolina, had found that very low doses of this same drug, propranolol, when given to patients with fibromyalgia or just localized temporomandibular pain, would reduce their clinical pain. This is data from that study, where this was a counterbalance single dose. Now, this wasn't a clinical trial, guys. This was a single dose given in a laboratory study only. But what we saw here was in the placebo session, if they had a pain rating, this was the average for the whole group, of 114, in the beta blockade session, it was reduced to a pain rating of 71. And we had a number of different events. They went from a supine to standing up because we were looking at postural hypotension in these patients. They continued to have lower pain after the rise to stand task. They also gave the simulated speech, another stressor, and they had lower pain after the simulated speech task. All of those were true on the beta blockade day versus when they were given only a placebo. Now, we've had a hard time getting these data published. I might as well tell you. So we were delighted when the animal data came out because we think then I can get these data in the literature. I haven't been able to up till now because this was too different when we collected these data. So I'm excited about that too. Also, among our patients were some who were involved in exercising on a regular basis, and some weren't. Those who did exercise on a regular basis showed some reduction in those ion channel receptor increases that we'd seen. But they were still higher than normal. And they also showed some reduction in their beta adrenergic receptors increases, but they were still higher than normal. This might be one way that exercise training helps reduce pain and fatigue symptoms if it prevents that increase from being quite so large and so dramatic. Maybe. Maybe. We also found that those patients who were higher in fitness, meaning those who exercise on their own more, they had lower cytokines. Interestingly, they had lower anti-inflammatory cytokines as well as lower pro-inflammatory cytokines. Both parts of their cytokine profile looked more normal than the patients who did not exercise. And this was especially true at the 8-hour post-exercise time point. I don't know exactly what's so important about that time point, but it could be that they are promoting further increases in some other changes. Okay, so I'm actually at the end of my study. I want to give you once again our caveat. This is the beginning of a very new research direction, but there is light at the end of the tunnel, and that way you'll remember who it was that did this work. And we have hope that these post-exercise blood-based measures can help us develop biomarkers that will give people credibility in terms of they have this disorder, this kind of thing. We hope we can develop these into true biomarkers that can be used for a medical purpose, and then maybe they will be clues to possible targets for future interventions. Thank you very much.